the 100th birthday of the famous book of Darcy Johnson. This book was is full of analogies, and it's very and Darcy Thompson knew that some of these analogies, he understood why they were analogous, and some of them were just visual analogies, and he insisted that it was a preface, and the whole book was a preface. And the fact that we are here shows that this inheritance has gone on all this during all this time, and now some of the things that were analogous at this time, now we understand why they are analogous. So there are many different ways of being analogous, and now you tend to speak of them as not analogies but convergences. And in biology, convergences are quite spectacular, and I will show you one. So in fact, there are physical convergences, that is phenomena that are driven by the same mathematical laws. They usually can form similar shape by growth, for instance, in the diffusive field or in the Laplacian field. They can also have similar shapes because of similar topological constraints. And then the biological convergences can also have one other source, which is, of course, evolution. So the functional convergence come from evolution, but they also can use the same physical self-organization to get these the same similar effects. So I'm going to speak about Fibonacci phyllotaxis. So it's a very old subject, and that plant growth is very slow, and the growth of its study is also very slow. So this study of Fibonacci organization has been spread out, and I will try to summarize some of its main breakthrough. So in fact, I will hopefully I will end by showing you that it's there's similar phyllotaxis in plants and algae. Okay, so the spiral organization of plants is apparently very well known since the Roman time. This is a bronze, Roman bronze, that is sitting in the Vatican Museum, and it is a perfect description of a pine cone. It is perfectly adequate. So either the Romans knew about its organization, or they copied very accurately the reality. And this, unlike modern sculpture showing pine cones. Okay, so the number of people who have been involved in phyllotaxis is enormous, and you can distinguish maybe three trends. One is to study the geometry of phyllotaxis, and you have heard the talk about it yesterday. Another one is dynamics of the apex, the growing part of the plant, and then there are all these studies that are more biologically originated, and they try to seek what is the physiology of this phenomenon. So the word phyllotaxis was introduced by Charles Bonnet in the 18th century. The first characterization of the phyllotaxis spiral were done by Schimper and Brown in Germany, and they pointed out that there were typically two types of organization of leaves around the stem. The one that is most common is spiral, so each leaf grows at a finite at a fixed angle relative to the previous one, and usually there are spirals. They can also be alternate with 180 degrees between two groups. And then there is another kind of organization, which is quite common in plants also, which are the whorl modes, where you have several leaves forming simultaneously. So for instance, there is a decussate mode where you have two leaves forming opposite to each other before the next two are tilted by 90 degrees. Okay, so this is just a description of what you see when you look at the stem of a plant. And this covers 99.999 of the plants. So the next important contribution is due to Bravais, so to the Bravais brothers in approximately the same period, middle of the 19th century. And what they did is to show that there was a relation between the organic. Oh, you don't see anything there. Okay, no, this is supposed to be a stem with leaves and the divergence angle. So they introduced this idea of divergence angle, which is the angle between two consecutive leaves, and they related it to this obvious phyllotaxis organization that you see in pine cones, where instead of linking each element to its nearest neighbor in time, you link each element to its nearest neighbor in space. So if you take this element, it will be on a spiral, which is called a parastixie by the botanist. And if you count how many of these spirals you need to cover the whole pattern, you find 8. And the blue ones, the other direction, you find 13. If you do the same with a sunflower, you also find two different numbers, and now there are 55, 89. So of course you know the answer, you know why. The remarkable feature is that these numbers, 5 and 8 and 55 and 89, are two consecutive terms of the Fibonacci series. And I must insist that it's not an approximate result, it's exact. When you count in a field, you can go in a sunflower field, you count the number of spirals and you find exactly Fibonacci numbers. There are a few exceptions which are from the Lucas series, which is built with the same rules as Fibonacci, but with initial terms 1 and 3. So there's exception about a few percent.
Sorry, I find the double number notation a little confusing.
No, I will come to it.
It relates to the triplet, yeah?
I will come to it.
Okay.
Okay, so in fact, the double notation is justified because, for instance, in this case, you clearly see that each element is squarish and it's connected to its neighbor. So in my figure, it's only two numbers are needed. I will come to it. Okay, so there is a relation between the divergence angle. So what the contribution of Bravais was to show that the divergence angle was very close to a value which is 2 pi multiplied by 1 minus tau, tau being the golden mean. And so there is a relation, well-known relation between the golden mean and the Fibonacci series, because the golden mean is the limit of the ratio of two consecutive terms of the Fibonacci series, as shown by Kepler, incidentally. Okay, so the work of Bravais was to show that if the angle is perfect, that is, has this value, then by compressing this pattern vertically, you change the parastixes, and you go spontaneously from parastixie 2,3 to 3,5 to 5,8 and so on. But this assumes that the angle is perfect. Okay, so that was a main contribution. Then later on, microscopy developed, and people from real biology, like Hofmeister in end of 19th century, looked at the structure of the apex, and he showed that the whole structure of the plant is originated in that region. So you have already seen some pictures. This is what you see if you open the bud of a plant. You find that there is a small dome, which is called a meristem, and at the periphery of this dome, you have primordia, which are the future. So there are little protrusion that will become, in time, leaves, flowers, and so on. So there's a more modern view of the same thing, where you see that it's at a multicellular scale, which is not always the case, because in some moss it is not. Okay, so the next contribution, which is a bit hidden in a book that is hard to get, is a contribution by Van Interson, who was a botanist, a Dutch botanist, who wrote this book. And he tried to understand, again by geometry, but the difference with the Bravais model is that Bravais used point-like structures, whereas Van Interson thought the primordia are finite size objects, And so you can relate maybe to the piling of this finite structure object. So he looked, for instance, something similar to what you heard yesterday, to the piling of disks on a cylinder that you unroll. So if you change, so this for very large disk, and it reduces the size of the disk. So you can see that here the paving is hexagonal, so you need 3 numbers to define it. Then it becomes square. You have only 2 numbers. Then hexagonal again, then square, then hexagonal, then square, then hexagonal. And he produced this remarkable diagram, phase diagram, where you have this divergence angle here as a function of a control parameter, which is the ratio of the size of the disk over the radius of the radius of the cylinder. Okay, so you see that for very large disk you are at 180 degrees, then you go down, up, and you converge to 2 pi multiplied by 1 minus tau. So in fact, this you can notice that this goes through transitions in these points, and in those points the pattern is locally hexagonal, and you go from one square organization to the next because some of the contacts get lost. So then he did also the same thing with growing disk at the tip of the plant and found similar rules. So that was a very important contribution. The next very important contribution is due to Turing, but unfortunately he died before publishing his result, and before writing them down. But there's very strong indication that they found self-organization in Fibonacci pattern in his morphogen, using morphogen diffusion. Okay, so in fact we undertook experiments a long time ago, so this, I'm a bit ashamed to produce some such old results with Stéphane Douady, and this started when he brought us a cauliflower, and we in fact revisited the hypothesis of Hofmeister. So Hofmeister, I recall you, he described the terminal bud of a plant, and he showed that there was a meristem, and he came out with very simple hypothesis that, okay, so the whole thing is growing, so in fact all the primordia are advected away from the tip. The meristem as a radius are 0, and he assumes that the primordia form with a fixed periodicity. They form at the periphery of the apex and in the largest space left available by the previous elements. So in fact, the remarkable thing is that those rules, that there's no biology whatsoever. It's purely dynamical system. So of course it's attractive to a physicist, and we thought if this is true, then you should see the structure in a system which is physical. And in fact, the remarkable thing about this Fibonacci organization is that they were known at that time only in botany, not in any other system. So we tried a experiment to see if these rules were sufficient to produce the result. So the experiment is rather simple and has absolutely nothing to do with the biology, of course. You have a small tank, you have oil in the small tank, and then you drop small drops of ferrofluids, which is a magnetic material. You have a gradient of magnetic field so that each drop, when it drops at the center, is attracted towards the border, so it will drift away from the center. And if 2 drops come close together, because they are dipoles, they repel each other. So with this, so there is a small pipette here, and you can drop the drops at a constant rate. And I will show you what you get. So you see the first drop goes randomly at one direction. The second one is repelled by the first one. The third one is repelled by the first and second. And as you see, it starts spiraling. So maybe you are not convinced. So we'll show you the next film, where the spiral is more sorry, no, it's not. Well, it doesn't matter. Okay, so this is the result of this experiment. This shows the organization they obtain after some time, and as you see, you have spiral organization, and you can draw parastickies, and there are 5 spiral in one direction, 8 in the other direction. So it is a Fibonacci organization. So why does it work? It works because the scaling is right, and in fact. You see in this experiment, as well as in the reality, in biological reality, you have only 3 relevant parameters: the velocity of advection, which is the velocity of growth in the plants, the time between formation of 2 primordia, and the radius of the apex. With this, you can do one non-dimensional number, and you take this in the experiment and in the real apex, and you find that the value of this dimensionless number is the same. So the geometry, this means, since these are lengths, Vt is a length, and R0 is a length, this means that the geometry of the system is the same, and that's why it worked. So then you can do numerical simulation. So you have a disk, you create new primordia on the periphery of the disk, they are advected away, they repel each other, you create them periodically, and you find that it does create spiral organization, Fibonacci-like. And if you plot all the organization that you get with a simulation, you get this. So this diagram is the divergence angle, the angle between 2 successive elements, and these factors, the dimensionless factor I've defined before, Vt over R0. If this is very large, each drop only fills the previous one, and you have 180 degrees divergence. And then you have a bifurcation here, where it goes either one direction or the other direction. This is this explains why the plants are randomly left or right spiraling. And then if you follow this line, you go. So here, if you look at the parasticki structure, it is square with 1, 2. Here it is hexagonal with 1, 2, 3. It becomes square again, then hexagonal again, then square, and finally you converge to 137 degrees, 0.5, which is the ideal angle. So this would be obtained. So the thing is that during the growth of the plant, when a sunflower grows, it starts at a large value here. So the first leaves are organized something like here. It's a little bit more complicated. I can discuss this privately afterwards if you wish. But roughly speaking, the first leaves are organized in this region, and then as the plant keeps growing, the ratio G decreases, mainly because the meristem grows. And therefore, you undergo a sequence of bifurcation. And when you reach the flower, you are at a very high orders of Fibonacci numbers. Okay. So there is, and maybe if I've there is an interesting remark, which is that it is somewhat surprising for a physicist to see a convergence toward a irrational number. So it is explained by the fact that in this organization, the system avoids rational number. So if you wish, if each element was only repelled by the previous one, it would be 180 degrees. That's where you get there. Then if it was repelled only by the 2 previous ones, it would go to 120 degrees. And then it also would go to, if it was repelled by the 3 previous ones, it would be 144 degrees, and so on. So you would reach organizations that would look like that. But of course, the elements repel each other, so they don't like to be radially aligned, and to avoid that, they have this repeated bifurcation that lead towards an irrational number. So it is really a very mathematical game. So there is a relation to quasi-crystal. As you know, there is a 1D model of quasi-crystal that was introduced by Katz-Andino in 1985. You take a square, quasi-crystal is an organization which is non-periodic, and it can be in 2D, as a Penrose lattice, for instance, but it can also be in 1D. And in 1D, you can obtain it by a projection from 2D. So you take a square lattice like this. In this lattice, you cut a band you first draw a line, which is at a rational angle from the lattice. So it passes by this point, and again by this point. And then you cut a band, and all the elements that are inside this band you project on this line. This gives you a repeated finite sequence of segments that are long, short, long, short, and so on. If you do it when the angle here is non-rational, then you pass only by one element and never again. And if you do the same projection, you get an infinite sequence of elements that is non-periodic. And therefore, you get a 1D periodic lattice. Now, it is exactly what you get in phyllotaxis, because here you have these parasticles. Around the circle here, instead of defining a band here, you define an annulus. And all the elements that are inside this annulus, you project on the central circle, and you get a sequence of short, long, short, which is, of course, periodic, because the circle is finite. But if you go to an infinite circle, you go towards a perfect quasi-crystal. This I don't have too much time to. So, of course, in all of this, I've forgotten about the world organization, the organization where you have two elements, and this. So there is another system which was proposed by Snow and Snow in 1951, both British botanists, and they said it's not necessary to impose that the formation of elements is periodic. You can just impose that an element has a finite size, and it forms as soon as there is enough space to put it. So you have a new dimensionless number, which is the ratio of the size of this element to the size of the meristem. So if you wish, it's a sort of piling system. So I don't have time to go into all the details, but what happens is that what you find is that there is spontaneously formation of a periodicity. So the periodicity is not imposed, but it is self-organized. And this periodicity can be with one single element, or with two, or with three simultaneously. And therefore, you recover a pattern where you have in black all the spiral modes that I've described before, and in orange here, patterns correspond to the world organization. In a way, these are rational organization, whereas the other one is sort of a irrational organization. I skip this because I have no time. It explains why in plants that are decussate, you can do an operation, single operation, that was done by Snow and Snow, and you have a transition in the later time. If you slit the apex of this plant in diagonally, you will get two new stems, and on each of these stems, the growth is spiral. So this means that it's not a genetic determination, direct genetic determination. It is self-organization. This was recovered in a numerical simulation. So returning to biology, so physicists are quite happy. They think that they have understood something. But in fact, the biologists are not happy because they think we have not understood why is there a stem to begin with, why is there a meristem, why is there a primordia, how do these primordia interact, and what is the evolution, what is the genetics. So they have a huge amount of questions, and so that leaves the subject again opened. So in fact, there are two. So the main question is, what is the physiology? So this connects to the present workshop. one of the hypotheses for the growth of plants and their anisotropy is mechanical constraint. Another important factor is auxin transport, which I will not describe. So, in fact, this led to a collaboration, international collaboration with many people from 4 different labs. And in this collaboration, as you can see, there is Rescue Daoud, who was very pivotal in the result. And where by applying forces onto the apex, you try to see if there is a reaction of the living tissue on the growing living tissue. And what you see, and of course I'm not going to describe because I have absolutely no time, is that you have orientation of the microtubules, and these will in turn orient the division of cells. So this is a purely biological approach, and what it shows is that mechanical constraints that are applied on the tissue act on the cell division, and the idea is that mechanical constraints that are due to growth without an external disturbance, the ones that appear spontaneously by the system pumping water out of the soil, they also create forces, and these forces orient the microtubule and therefore make the system spontaneously anisotropic. So, okay, I will skip this. So I'll come to the thing that is linked with my talk. So after you have done this biological approach, you feel, yeah, but what was it useful to have this physical understanding of Fibonacci organization? And this is so the result of this approach by physicists is to say that it is something that could be very general, and that it is a spontaneous self-organization that doesn't need to be implemented in plants. So in fact, it predicts the possibility of a convergence, and we were happy to find that it is indeed there are such convergence. When you go to the seaside in France or in Great Britain, the most common alga, brown alga, is fucus, which has this shape, and you know it's characterized by this systematic tip splitting, and you see it everywhere. But now if you go recently, you will find that there is new alga that I didn't know when I was a kid, and that is everywhere, and it's called sargassum. Sargassum, the reason why it was not there when I was a kid is that it is an alga that has invaded the coast of Great Britain and France and Netherlands, everywhere, Poland probably. It came with oysters. We imported oysters from Japan, raised them, and in these oysters were these seeds for these algae. And they have been proliferating, and now they are everywhere. They're very successful.
Not quite everywhere, I think. I haven't seen it.
Well, maybe, yeah.
I will look into this.
Yeah, you must look. Because they change quite a lot with season. So you have to look at various seasons. They are more visible in summer than in winter.
Give me a project.
Okay, so they have this. The spectacular aspect is that they don't really look like algae. If you look at them, they look like plants. You have a stem, you have branches, you have small leaves, you have reproductive structures that are there. It really looks like a plant. So if you go further, you can look at the tip here. And if you look at the tip in detail, this is what you see. And you see you have these pseudo leaves, which are not leaves. The stem is not a stem. The leaves are not leaves really. And you can number them, and they form a spiral. And you can measure the angles between successive elements, and you find that with a good approximation, it is close to the ideal Fibonacci order. So in fact, so we were all excited, and then we went into the old books, and as usual, we found that it was known. It was known in a book by Church in 1920. And it's just a sort of half a page where he shows one drawing, which you can't see, but unfortunately. And he says it looks like a Fibonacci organization. And he says, but he adds, it is a Fibonacci organization because the algae are the ancestors of plants. And this means that the Fibonacci organization went from the sea to the land. Okay, and this is wrong. This cannot be, because now we have a phylogenetic result showing how these different clades are related. Plants are here, and brown algae are here. They have diverged from each other 1.8 billion years ago. That is to say that we are more related to fungi than brown algae and plants are related together. Okay, and they diverge at a time where both clades were monocellular. So this means that all the pluricellular structures have appeared independently in plants and algae. So we went back. So we went back to studying to looking at the algae more seriously. So the pictures are not too good, but this is the apex of Arabidopsis thaliana, and you see the meristem, parastechis, the spirals, and so on. This is the same thing for Sargassum. And as you see, you can define, you can see the primordia, you can see a sort of central meristem, primordia, organized spirally. The main difference is the size, where the size here is larger, 10 times larger than in plants. You measure the angles at the tip, and you find that in both cases it is somewhat similar, with some spreading, of course, because algae are very soft and not very easy to manipulate. But there are differences, though. For instance, the meristem, instead of being a stable dome, is centered around a pit. And in this pit, you have so this is the meristem of a plant. This is the meristem of the alga. It has a pit here. The pit is centered itself on a big cell, so the structure of the meristem is quite different. But yet, you find the same organization. So in fact, you can go further and look a little bit more in general. You find that algae, in fact, have two sorts of configuration. One is with tip splitting like this. One is with very anisotropic growth like this. And you find this also in red algae. This is a different branch. These are brown algae. These are red algae. Doesn't show that they are red, but they are red algae. And you see that you have two regimes, one with tip splitting and one with growth with axis and spiral. So you can go back to plants. So in plants, you know, most plants grow with axis and with spirals and so on. So you find it in moss, in ferns, in normal plants. And are there isotropic growths in plants? Well, I didn't know, but there are. There are very primitive plants called liverworts, and they have a dichotomous type of division. So of course, so I will skip this because I'm out of time. So why is it this analogy is interesting? One reason is that, okay, so the plants and the algae have found the same self-organization system, probably very independently. But this self-organization system has a major advantage, because you see that this alga looks quite miserable at low tide. But if you look at high tide from below, you see that because it has these stems, it forms like bushes that grow quite high. The length of this alga can be several meters high. And so it first catches light better, because these are photosynthetic structures. And second, it spreads its seeds, well, it's not seeds, but it's a reproductive structure, in the current, in region where, away from the boundary layers. Same thing as plants. You know, when plants grow, they grow up, and the higher they are, the easiest it is for the seeds to get away. So, in fact, conclusion is that it seems that the two clades have found the same system of organization, and the evolutive advantage seem to be the same. Thank you very much.
Have some questions?
Some plants have a double Fibonacci series.
Yeah.
How do you accommodate that?
You find it in the simulation as well. It is a decussate mode that goes spiraling. And it's you can actually do, maybe we can discuss it in private, but in sunflowers, so if you have many sunflowers, 95% of the sunflowers are real Fibonacci, 2.5% of Lucas series with 1-3, and 2.5% are.
So in that case, it's some sort of an accident of how
it is initially. It is all these anomalies are initially determined. It's at the first bifurcation when the plant is still very small that it bifurcates, and then it keeps growing in the same way.
Is there an equal distribution between the left prevalent and right prevalent?
It's equally probable because it depends only on one event, which is the initial breaking of symmetry. So it is with equal probability left or right, which doesn't mean that all spirals in plant are equal. These are, I'm speaking of these phyllotaxis spirals. There are other spirals in plants which have a handedness, like for instance some wines or things like that. But this is not related to phyllotaxis.
In your magnetic experiment, is there a limit in which the rate matters? The rate at which you drop you add?
Yeah, you can't go very, very far because drops start falling onto each other. So it's a practical limitation that if two drops fall onto each other, they coalesce, they form a big dipole, and the experiment is
But apart from that, it doesn't matter.
no.
All right, let's thank you again.